Okay, so thank you everybody attending this talk. I want to thank Veterinary Bioscience Institute and Lab Roots for giving me the opportunity to discuss what I'm going to present to you. I think it's an interesting topic, and the talk is going to be around rodent husbandry and the environment we keep the animals, and the impact that this may have, and this is a very important thing, may have on experimental results. So let me start with this chart representing the logic or the mechanics, if you will, of a typical experiment. So we start with a biological question, a biological problem, and then we formulate an hypothesis, and then design experiments to test that hypothesis. Then we run the experiments with our treatments, our controls, and then we collect, we get the data, the results, and then or through the analysis of those results, then we find a conclusion to answer that biological problem or question. So ideally, the difference in the results that we see should come from the treatment, but this is not always the case, right? And this is basically because there are a lot of factors that can influence the data that we collect from an experiment. These factors can be due to the experiment itself. It can be experimental factors like the experimental design, the treatments that we apply on the animals, how we apply those treatments, how well trained the people running those experiments are handling animals, data collections, errors from data collection, or even the equipment can be a factor influencing. So if the equipment is not calibrated, things like that can influence those results. But more important for our discussion today are the non-experimental factors, right? And I have divided those non-experimental factors for our discussion today in intrinsic or animal-related factors, the physical environment, and the non-physical environment. Let me also say that the control that we choose for the experiment is extremely important. That's not the discussion today, but the control is going to have an impact in that difference because we are comparing the treatment with the control. So we've seen very high-level overview of these factors and how diverse they are. But let me remark here that all factors that can impact laboratory animals may have, and again, I would like to remark that, may have an influence on results. Obviously, if these factors affecting the animals are not fixed, then this is when they become variables, and then this is where the problems may start. So let me expand a little bit on non-experimental factors. I have divided these non-experimental factors into intrinsic or animal-related factors, like the age of the animals, the gender of the animals, the genetic background. We all know how important the genetic background is. The microbial flora of the animals. And this is an expanding field at the moment. The health status of the animals, that is obviously an obvious influence on the results. The physical environment also have, and this is what we're going to discuss tonight, has an impact, and I'm going to give you examples of this impact. But the variables, the main variables of this physical environment can be, temperature, humidity, noise, light, also very important for rodents, the circadian rhythms, and this is in some cases this is classified, or I have seen this as an animal-related factor, and in fact it is an animal-related factor, but the environment has such a powerful influence on circadian rhythms that for the discussion today I'm going to consider that as part of the physical environment of the animal. And obviously the non-physical environment, so these parts of the environment that are non-physical, actually can or may influence the results. And I mean, things like the interaction with other animals, even same species or at different species, the husbandry procedures, the handling of the animals. This is very very important, and I'm going to show you in a minute a few examples to illustrate this. Also the staff, we have husbandry staff, animal caretakers, scientists, technicians performing experiments. All that is gonna influence the results. It's gonna have an impact on the data that we collect from the animals. So these non-experimental factors we can group in the ones that are animal-related, and the rest is the environment, and that's going to be the discussion today. We know, and we all know, that the environment have an impact on reproductive parameters, on animal welfare, on experimental results, which is the subject of the discussion today. The reproductive parameters in rodents are very well characterized. So it is easier to see a deviation or to identify a deviation in these parameters, and then we may be able to link a problem in the environment with a reproductive problem. Also, animal welfare we can also assess and also, not easily, but at least be able to link deviations in this animal welfare with the conditions of the environment. But it is much more difficult to link deviations or influence on the experimental results of this environment, simply because of the very nature of the of research. In many cases, we don't need to, we don't have anything to compare with. So it is a lot harder to identify the influence of the environment in the experimental results. And this impact or this influence of the environment is critical for the discussion of the results, as I just said a minute ago. And so that's why it's so important, the standardization of the animal model, the environmental parameters, and the experimental protocol. And all these will make our experimental results reproducible, which is, the central subject of science, the fact that we can reproduce those results. So the impact of the environmental conditions on the response of an animal model, we could describe using this formula, if I can put it that way. So the genotype of the animal plus the environment plus what I am studying, what plus the test, will give us the phenotype. I said the genotype plus the environment plus the test, and this is not entirely true, because it's not one plus the other plus the other. It's a lot more one influencing the other, So it becomes a lot more complicated. But anyway, let's focus on the environment. So an adequate environment for the animals must provide socialization for the animal. This is critical for social species like rodents and many other animal species. Must provide to the animals the ability to exercise, also to promote their physical development, the behavioral development. It is also paramount animal welfare and an optimal health status. So these are, features of an adequate environment. Let me remark also here that we start from minimum standards that are described in the laws that regulate animal use. So an adequate environment is also described in the current regulations that we apply. It can be the guide in the US, and here in Europe much more the new directive. This set of regulations give us a minimum standards for the environment of the animals. Let me describe a couple of things before I get into the examples of how the environment can influence the results. Just to make this distinction, and I'm sure you all know, but the primary enclosure, or what we describe as the microenvironment, and the secondary enclosure, or the macro environment. The primary enclosure is the cage where we keep the animals, while the secondary enclosure will be the broader environment, the room, usually the room where we keep the animals. It is very important to note here that it's a combination of both what really defines the quality of the environmental conditions. So let's start discussing or getting into a little bit more depth, talking about the microenvironment. There are many parameters that can affect our research results. Some of them, and I put these here in no specific order, but available space per animal is a very important parameter. The number of animals per cage, that's also critical. I will give you example of individual versus group housing, and how these influence research results that you wouldn't imagine if you didn't do the test. The cage size, the environmental enrichment, another very important factor, and the quality of the materials and the equipment. This can also have an impact on the experimental results. So just to illustrate, and I'm going to give you examples here. So the purpose of the talk is not to discuss the results that I'm going to present here. These are just examples. But I think that they are very illustrative of how these environmental parameters will influence the research. So this is a study where they compared individually grouped, individually housed mice with group housed mice in pairs. Right? So I don't know if you can see very well here, but this is they measure, and these were measured by telemetry, the heart rate and the body temperature. Right? And this and they measure that during periods of low, middle, and high activity. Right? So where you see the differences here with the stars, those are significantly, statistically significant differences. So just the fact that the animals were singly housed or in pairs is got an influence on the heart rate and the body temperature. This is also part of the same study, and it's interesting that the activity of the animals is also different. These are the 24-hour day. Then they follow the activity, and you can see here that in animals that are housed individually or housed in pairs, you can see that the activity is different. You see high activity here, while the animals housed in groups are in middle activity mode. Then when they become highly active, then the singly housed animals are not so active. In this 24-hour period, the lights will go on like around here. So you can see that the pattern is also different. There's a period of low activity here for the group house or the animals housed in pairs, compared to interaction of these low activity by periods of middle activity in the singly housed animals. So this is just an example of how something so simple as housing the animals singly or in pairs will impact parameters like activity, heart rate, or body temperature. This is another example, and I think it's very illustrative of how housing mice in groups or individually will influence a different study. This is a force streaming test, and they test a compound to see if they have antidepressant activity. So you can see here in white the animals housed in group, and with this shading light gray, animals that are singly housed. Right? So if you compare the control with two different doses of that compound, and look at the duration of the immobility, which is what we measure in the force streaming test, you can see that the results for the group housed animals indicate that there is an activity, there is an effect of this compound on the duration of immobility. While if you compare with the singly housed, there's no effect for that compound, right? You can also imagine that if we had a mix of singly housed and grouped housed animals, this will make the whole thing more difficult to interpret. Let me give you this another example of a different parameter in the microenvironment or in the environment of the animals. We need to handle animals, for the cleaning, for the experiments. That's unavoidable. But let me illustrate the effect that handling animals may have on experimental results or in physiological parameters. This is a study where they compared mice that were changed. The cage was changed once a week or every other week, bi-weekly. So every 7 days, every 14 days. There are variations that they're not statistically significant. That is also important for experimental results, and I will discuss that later, simply because they are introducing noise in the analysis of the data. But if you look at this specific parameter, which is plasma cells, you can see here that just by changing the cages once a week or every other week, we see a difference which is pretty much double the parameter. So it's got a clear influence on physiological parameters. This is another example just to illustrate a little bit more how handling, in this case, pregnant rats can influence. In this case, what they measure was the fetal body weight. So they stressed or they handled their rats the third gestational day, the fifth, or the seventh, and these are the controls. And they found that when they restrained the animals, the pregnant rat, just for a minute on gestation day 7, then when they collect the fetuses on gestation day 18, there will be a difference with what we see on controls, right? So it's just is not a big alteration. So we're just restraining the animal, but the effect we can see. This is part of the same experiment, and you can see some other parameters here. And I don't know if you can see it very well. I will try to highlight some of them, but they handle the animals. They restrain the animals on gestational days 1 to 4, 5 to 8, 9 to 12, or 13 to 16, and you can see. Again, it's not the purpose of this talk to discuss what's going on here, but you can see difference, right? See differences here, statistically significant differences here, there, more there, right? So just handling the animals will have an impact on the physiology of the animals and therefore on the results. But the microenvironment is a lot more complex, and we're usually looking at a combination of different parameters. This is another study just to illustrate how the combination of these parameters can influence, in this case, just reproductive parameters like the pre-wean mortality in mice, in this case, and production of animals, mice wean per dam. So what they compare here are pairs and trios, and these animals were kept in IVCs, in individually ventilated cages at 30 air changes per hour, 60 air changes per hour, renovation of the air in the cage 60 times per hour, or 100. And what they did is to change the cages once a week, every other week, or once every 3 weeks. So again, we're not going to get into the into details, but you can see that there are differences looking at the different combinations, right? So in this case, the renovation of air, the environment in the cage, and the handling has different impacts in the breeding performance of these animals. By the way, it looks like one of the best conditions for keeping these animals could be around 60 air changes per hour, and maybe handling the animals every other day. So that's anecdotal. I think what is relevant here, and what I want to bring here, is that there is an effect. There is a combination of effects. Let me touch a little bit on environmental enrichment. There is a lot of discussion on environmental enrichment. So I'm not going to go into details about what whether environmental enrichment is good or is bad, or is good for certain experiments, but not for behavioral studies and things like that. What I would like to bring here is that environmental enrichment is another factor that may alter or may influence our experimental results. Let me make it clear with this example here that animals seem to prefer an enriched environment. Right? So this is an experiment where they offer the animals a slightly enriched environment, like just the cage plus the grid and nesting material, or just simply the box, just the cage. So this is the time, the dwelling time, and it is clear that the animals prefer to be or to dwell. They dwell more in the enriched environment. So, well, that's the preference of the animals, but the fact is that environmental enrichment may have an impact on experimental results. And again, it is important here the word may have, because if the environmental enrichment had an influence that is always of the same magnitude in the same direction, then we could, live with that. But it looks like it's not always the same. For some parameters, it's very important. For some others, it's irrelevant. For some studies, it may have a huge impact. For some others, it's not so relevant. This is just to illustrate something that we all know, that environmental enrichment has an effect on mouse behavior. It's pretty obvious that an enriched environment will have an effect on activity and playing behavior, and we can see this here. I don't know if you can read this, but this will be a standard cage, an enriched cage with nesting material, and a super enriched cage with additional complexity in the cage. And you can see here that the differences are statistically significant. Part of that study as well was to measure some physiological parameters, and once again, it's not so important here what those parameters were. What is more relevant is that they found differences, statistically significant differences in those parameters depending on the standard cage, enriched environment, or super enriched environment. So for this study, they found these differences, but let me bring here another study where they measure hematological parameters like white cells, red cells, hemoglobin, or the hematocrit of the animals. And in this case, we using different strains like A strain, Black 6, or Bulb C. And in this study, what they could see is that the effect of the genetic background, the strain, was significant in many cases, but not the housing, not the difference in the housing conditions, and enriched versus non-enriched. Right? So this is again to illustrate that for some parameters it may have an impact, for some others it may not. And let me just bring another set of results here. It's part of the same study, but this is comparing body weight and weight for different organs, kidney, liver, spleen. Again, they compared using different strains, and they couldn't find obvious differences or statistically significant differences here. But what is relevant here, and the point I would like to make, is that for many of these results, they saw that the animals in the enriched environment had a greater variability. Like, for instance, if you compare here, that's the coefficient of variation. If you it is consistently, I would say, higher in the animals in the enriched environment as compared to the animals in a standard environment. Like you can see here. So it looks like in this case, and for this particular study, and for this particular parameter, which is body weight and organ weight, what environmental enrichment is additional variability. Let me continue with the microenvironment and make a comment, because especially for rodents, it is very, very common to use individually ventilated cages, right? So this is an additional factor that will influence the microenvironment of the animal. Individually ventilated cages is a great system to keep the animals. I mean, it can reduce the pathogen spread in case of an outbreak. It certainly allows better control of temperature and relative humidity. Something that is also very important, these IVCs reduce gas contamination or ammonia production in the cage. And also let me remark here that they can be very efficient in reducing our exposure to allergens. So that's very good. But I just said that they reduce the production of ammonia, for instance. I'm just giving you examples. And ammonia, it's an environmental factor that will have an impact on the physiology of the animals. So I just put here a couple of examples of acute exposure or chronic exposure, and you can see that exposing the animals to 50 parts per million or 100 ppm for 2 hours or 3 days, it's got a physiological impact. It's got physiological consequences. Chronic exposure as well to 25 ppm, which is very low, for 6 weeks, it's got a physiological impact. And exposure to 50 ppm for 3 weeks, it may cause immunosuppression. So again, IVCs in this case can be modifying the environment. So if we compare animals, running the same experiments, and we have animals in conventional cages, and we're comparing with animals in IVCs, they're not in exactly the same environment, even if they're in the same microenvironment, in the same facility. So that's something to take into account as well. So this was just a few examples of how the microenvironment can be altered by environmental factors, and therefore the experimental results. I'm going to discuss now very briefly the microenvironment, and I'm going to briefly discuss and give you some examples of how parameters of the microenvironment can influence experimental results. I'm going to talk about ventilation, light, and noise because they're the most obvious. But let me first show you a few examples of differences found in experiments that can only be attributed to just a different lab. So in this case, and again, I don't think you can see the picture very well, but I will try to comment to describe a little bit the experiment. You can see here black red and yellow bars. So the black bars are the results of the same experiments with the same animals run in a lab in Albany, in New York. The red is in Edmonton, in Canada, and the yellow one is in Portland, in Oregon, I think it is. So again, like the other examples I brought, I'm not gonna go into detail. These were behavioral studies, but what is really relevant here is that a lot of the parameters, of the experimental parameters and animal-related parameters were standardized. They say rigorously equate in this study, and despite this standardization, there were differences across the labs. Right? And it is very interesting, the comment that they made here, that for some of these tests, the magnitude of the genetic differences depended upon the specific testing lab. So it's not just the experiment, it's not just the animal, it's the testing lab. I think this is very relevant when we try to reproduce experiments. There is an intrinsic variation that depends just on the lab. Let me give you another example. This is a communication. I don't think it's published data, but these are 2 rat strains, and these strains were kept in an animal facility, and the number of the percentage of productive matings for each one of these strains were 60 and 40%. It's not very good, but that's how it was. So there was a refurbish of the animal facility, and then the colonies were taken to a different room. And you can see that just by moving the animals to a different room in the same facility, the productive matings, the percentage of productive matings just changed. Again, it's just exactly, they continued doing exactly the same, but in a different location. And just to finish with these comments about just the place where experiment takes place, just a comment that or I want to bring here the variability, like in reproduction, just with seasons, right? So simply because of the place the Earth is spinning around the sun, we may find differences in reproduction in mice. Right? So let me just make a few comments about ventilation. Is probably one of the most important things in an animal facility. It provides the air, the fresh air, the oxygen. It is the main means of for reduction or elimination of contaminants, ammonia, allergens, microorganisms. It is critical for temperature control, and also helps with odor control. There are standard agreement for areas or for what the parameters, the ventilation parameters in terms of temperature, humidity should be. And if you can, if you see here this chart on your left, this is a chart of again, it is very small, but I think that you can see here, this is the concentration of ammonia, and these are air changes per hour. So the fact that we tend to fix air changes per hour at 20 is simply because we guarantee a nice environment, a non-offensive environment in terms of ammonia at that level of air changes per hour. So the non-offensive level will be around here. So this curve is for a room that is optimally designed. So the ventilation it's well designed, it's very efficient. This one here is for a non so efficient ventilation. But you can see, so with a very efficient ventilation at about probably 10 air changes per hour, we should be on this comfort zone. While for poorly designed ventilation, we will need to go to 18, 20. But in order to guarantee that the environment is right, we pick those 20 air changes per hour. Pretty much the same thing applies. That comfort zone for relative humidity. If you see the chart on your right, the graph on your right, this is 10% humidity. This is 90% humidity. These are different parameters as bacterial growth, the spread of fungi, mites, and things like that. And we've got this comfort zone about around 50%. So anyway, that being said, let's talk a little bit about the temperature. But let me point out first that we keep the animals around maybe 22 degrees Celsius because of those regulations and because it's comfortable for humans. But the comfort, the temperature that is thermoneutral for mice, it's around 30 degrees, right? So that's important to keep in mind. Again, a chart or this is actually an older study, but I think it's very illustrative. And this is an experiment where they measure the declining serotonin. Again, what they measure in the experiment is not so relevant for our discussion. What is really relevant is that they measure that parameter, that experimental result at 9 degrees, 24 degrees, or 34 degrees. This is the environmental temperature. And they found differences in two of the experimental parameters, right? So just the fact that the animals were kept in an environment of 9 degrees, 24 degrees, or 32 degrees, this had an influence in this, serotonin decline constant. I think that's pretty illustrative. Light, again, there are obviously a lot more studies on light and how light intensity and cycle has an effect on physiology and morphology. This is actually very well characterized for albino rodents, but also on the behavior of rodent species. It is probably a very good thing that we keep a dual intensity in the room. And let me point here that 350, that's the maximum light intensity. 350 lux, that's the maximum light intensity for rodents. Again, before I make further comments on light influence, let me see, because we tend to think that if the environment is okay for us, it is okay for the animals. But this is the relative sensitivity of the human eye and the mouse, and they don't see. I mean, it's well known that they, beyond this point of the spectrum they can see, so they can see red light, and this is under debate. But anyway, and they can also see a little bit on the ultraviolet area. But let me discuss this, and the point I want to make here, and this is my own data in a room that we kept just mice. So the intensity in the room was 150 lux. So you may think, well, that's a very nice lighting for a rodent room. We kept the animals in IBCs, so you can see here the intensity of the light inside the cage in these IBCs. So depending on the row and the column, the position on the rack, you will have intensities, light intensities from maybe 22 lux to maybe 2, right? So that's 10 times more. So there's a lot of variability here. But if you take the cage from the rack, we measure the light intensity inside the hood, the laminar flow hood, and we had 2 hoods in the room. In one of the hoods, the light intensity was over 400 lux, while the other one was almost 1,000, right? And then we also measure the light intensity when you open the cage. So in the cage, when you remove the lid, and in this case it was like almost 800 lux, but in this case, in the second hood, was over 1,000. And the third thing we measure was the light intensity at the level when you transfer the animals from one cage to the other, and you can see the intensity of the light here. So in less than 1 minute, you take an animal that is probably sleeping at 2 luxes, then you take it into the hood, open the lid, pick the mouse, and then put it back again. So that's stressful, I would say, right? So again, light is a very important factor, and light cycle is very important as well. Let me. I brought this example here, and in this study, they transferred tumor cells. They implanted tumor cells in buffalo rats, and these rats were kept in 12-12 day light dark phase, light contaminated dark phase, or in constant light. And depending on what environment the animals were kept, the weight of the tumor, and then the growth of the tumor was different, right? So there is a direct impact on the light cycle in tumor growth. That's affecting the results. Just another comment on circadian rhythms. It is so circadian rhythms is intrinsic to the animal. It can be affected by the light cycle, but even if it's not affected, if it's stable, just the fact that they have this rhythm, this gives us variability, right? So we know that rodents are more active during the dark phase, and this has effect on the physiology, like for instance on their metabolic rate, and this may mean that the drugs that we give to the animals or the effects of those drugs can vary quite a lot depending on the time of the day they are administered. And I want to give you or to show you this example here, and these were nude animals that were implanted, that were injected with tumor cells, and what they measured here were cells in S phase, so DNA replication, and telomerase activity, and you can see that depending on the time they collected the cells, there were, maybe very significant variations, right? So it is good. I think it's good advice to repeat the experiments and about the same time of the day, especially if what we're measuring can be linked to circadian rhythms. Let's discuss a little bit about noise. And again, like with the light, I would like to remark that what we hear there is not what mice can hear, right? So the fact that we say that a background noise of 50 dBA is okay, this is not. We're not taking into account ultrasounds like from here, so we may be disturbing the animals with ultrasounds. Again, just another example to illustrate how noise can have an impact on the physiology of the animals. This is a very old study for, I don't know if you are familiar with this silent alarm, but this was a study to show that the silent alarm had no effect on the animals. So so the experiment was to trigger the fire alarm for 1 minute 3 times a day for 4 days, and when they did this here, they could see a variation on the number of cornified smears cells. And this was in rats, right? So right there we see an effect compared to control animals. So that's a clear effect in the physiology of the animals. I also mentioned at the very beginning that the quality of the equipment can have an impact on on experimental results. Let me just bring the case here as an example of the famous bisphenol A. Right, this is the normal component of the plastic compounds that we use in many of the cages that we use to house the animals. But I'm sure you have seen things like that, the degradation of the plastic material, and this is telling us that the bisphenol A is starting to be released. So again, to illustrate the effect that this may have on experimental results, this is an example of the fold increase in chromosomal abnormalities measured before the cages were damaged during the period when animals were exposed to this bisphenol A, and then right here they started to replace the damaged cages, and you see that after the replacement, the chromosomal abnormalities came back to the original status. Okay, so I made comments and gave you examples of how the physical environment and the non-physical environment, remember the handling, I think, can influence the experimental results. Let me finish with a comment on also how the environment influences the 3 R's, especially the reduction. And it's simply because this variability that the environment is introducing, it usually causes statistical errors or deviations, false positives, false negatives. Because of that variability, we may need to add more animals to our experiments to achieve statistical significance. And even worse than that, I mean, we can find that our experiments cannot be repeated by ourselves, by other groups, or even much worse. And we may find that experiment after experiment, we find inconsistent data. So the experiment that we ran 3 months ago, we obtain data that are not consistent with what we are obtaining today, and this creates, confusion, frustration. I'm sure you all been through that, or know someone that has been through that. So okay, so that was all I wanted to bring for the discussion today. I thank you for listening, and if you have questions, I will gladly take those questions and bring them for discussion. So thank you very much. Okay, so I have a question here asking whether a copy of this presentation will be posted for download. And I think so. I don't have a problem with that. I mean, most of the information that I presented here is not my own information. I results from other people. I just collated everything to illustrate how the environment can influence experimental results. So this is available for download. I'll make sure that you can download it. Okay, if you don't have any more questions, so thank you for listening and good night.